Hey, so tonight it's my pleasure to welcome Dr. Bridgette Clarkson. She is an assistant professor in the botany department at UBC's Faculty of Science, specializing in seaweed biodiversity and systematics. Bridgette grew up in the Comox area of Vancouver Island, where she spent much of her childhood at the seashore, dragging home dead creatures deposited by the tide. I did read that on one of your sites, Bridgette. I didn't make that up. It sounded like a great childhood. She completed her PhD in New Brunswick, studying the diversity of red seaweeds in Canada, even discovering a few new species. Bridgette co-authored the book Pacific Seaweeds with Louis Drouel, as well as authoring the pamphlet field guide, Seaweeds of the Pacific Northwest. Oops, right. She is a mother, a talented photographer, and an all-around lovely person. So all those of you with the good fortune to be registered for the field trip with Bridgette on Saturday, you're going to enjoy a wonderful and enlightening outing. And with that, I'm going to turn it over to you, Bridgette, and I will get out of the way.
So first, I'd like to express my gratitude at being invited here and getting to spend some time with, gosh, it's like something like 50 people on this call right now. So that's wonderful to be part of a group spending their evening talking about seaweeds. And so tonight, I'd like to share a couple of stories in celebration of the seaweeds of the Northeast Pacific, and also some of the work that I do here as a professor of teaching at UBC, trying to get my students engaged, or the wonderful work that my students do, building materials to kind of put the word out there about how awesome seaweeds are. And thank you, Joan, for the land acknowledgement. I did also want to acknowledge myself that I'm joining you today from the traditional, unceded, and ancestral territory of the Musqueam people. This photo shows my office is actually just behind those trees on the right side of the picture there, or left side, and that's where I am now. I've been very privileged to get to participate in some initiatives through UBC that are in collaboration with the Musqueam. And it's really transformed my approach to my work in collecting seaweeds and thinking about my impact on the land and what I teach my students now about land stewardship and ethical collecting and many other things. So I feel very privileged and acknowledged that I am on someone else's territory doing my work. So Joan mentioned that I'm born and raised here in British Columbia. I grew up in Comox, or on the traditional territory of the Comox First Nation. So I'm from Vancouver Island. I was very lucky to grow up right on the beach. I didn't realize it then, but I do now, how incredibly rare that opportunity was. And I did drag many dead things home as a child. I still do it now. I just found something dead in my bag earlier today that I had to air out the office. And I caught this seaweed bug that really put me on this path. As a student, when I was at UVic as an undergraduate, I took a seaweeds course with those two fellas down there. That's Doctors Louis Drouel on the left, and then Gary Saunders is in the middle. And that's myself at the 2018 Phycological Society of America conference that I co-hosted as part of a team here at UBC. Gary was my academic advisor, so he was my PhD advisor. Louis, who is a retired professor from Simon Fraser University, he was Gary's advisor. So Louis is essentially my academic grandfather, and he's been a friend and a mentor ever since I took a course with them back in 2004. So it's been 20 years that I've been on this path, and it was transformative to have the experience of being in Banfield and learning from these two gentlemen. So I'm going to share a bunch of things today, but really where I come from now, as Joan mentioned, my background is in systematics, so trying to understand the relationships and classification and natural history of different kinds of red seaweeds was my specialization. But since I came here to UBC, I've kind of put on a different hat as an educator about algae. So I work a lot with how do we teach people better about all this fascinating stuff that we know about seaweeds. And so I do things like publish lesson plans and activities, among other things, that are about getting people involved in and excited about seaweeds and algae. I'm going to take a step back here, though, for a second, because I've used algae a couple of times, and I'm also using seaweed. So I want to clarify these two important and often confusing terms. So I have a set of images here. They're labeled A through H. They show 8 different individual organisms. In one photo, H, there's actually 2 things. I'm talking about the orange fuzzy stuff on that western red cedar tree bark. I would like people to, in the chat, if you want to, go ahead and tell me which of these you think would be called algae. And you can think of why too, but just off the top of your head, which ones would you call algae? But don't type it in the chat yet. Type it in the chat, but don't hit enter. We're going to all hit enter at the same time. Okay, I'm going to give you a minute to think about it. All right, if you put one or more letters in the chat, go ahead and hit enter and let's see what people say. Okay, whoa, look at those responses rolling in. B is a very common one. A couple folks said C, G. Okay, yep, G is another one that looks right off the top of your head. F. Oh, I should have said E has 2 organisms too. Pardon me, that was confusing. Ooh, definitely people think B a lot. Oh, Kira said A. Hello, Kira. It's controversial. I like your stance. Okay, B, D, G, and H. Okay, so there's a smattering of different ideas out there, but what would be considered an alga? Alga is a singular for the word, algae is plural. I'm sticking with it too. I like what you're thinking there, Kira. I'm sticking with A. All right, is anyone brave enough to turn their mic on and tell us why you chose what you chose? Sheila, I know your mic works.
I think Kira should tell us why A.
Yeah, Kira, tell us why A. I can tell you why A in a minute, but also why not A. All right, that's fine. Here, people, I'm going to give you what I would consider to be, that's okay, your mic's not working. I wondered if it was something like that. All right, so of all of these illustrious creatures that I have here on the slide, I, as a phycologist, a person who studies algae, this is what I would choose as belonging to the algae. And so this is an exercise I go through that Got Algae is all about this. It's like, what are algae? How do we define them? How do we understand this diversity? Why are they so diverse? And so B, a lot of folks got that. B is actually Salish, one of the species of Salish. It's named after the Salish Sea. It's the genus that I was very privileged to be able to document and describe for the first time as part of my graduate work. So that's our Salish Sea seaweed. D, I think most folks got, that's the bull kelp. That's one of those classic exemplars. E, that bumpy little bubblegum smear on the limpet shell, that's a crustose coralline growing on top of a limpet snail or a limpet shell. F is a kind of phytoplankton that belongs to the dinoflagellates. G, many folks got, that's our classic sea lettuce. So B and G have this classic seaweed body, these leaf-like blades. And then H is actually a kind of terrestrial algae. It's a green alga named Trentifolia. It's all over UBC campus and around Point Grey. It's really flourishing at the moment. And so all of these would be considered algae. C, which is a larval form of a crab, that's an animal. So we don't consider algae to be animals and vice versa. And then A, okay, so A is a skunk cabbage. Why would A maybe be algae and maybe not? It comes down to a couple of things here. So maybe this is preaching to the choir and you all know this. If so, I apologize, but this is often a confusing, oh, H is not a lichen. Sheila, H is often mistaken for a lichen. It is not. It is purely alga. It is a green alga. It has evolved to have additional pigments, basically a sunscreen for itself. And that's why it looks so orange. It's because it has a ton of carotenoids that are masking its chlorophyll. So algae are usually photosynthetic, usually aquatic. There's exceptions to all of these features that algae have, but the vast majority of them are photosynthetic. If they're not currently photosynthetic, they had a photosynthetic ancestor. But things that algae do not have, we consider them to not have seeds, roots, or flowers. They may have structures that are performing a similar function, but they don't have true roots. They don't need to suck up water from their substrate because they're often growing on rock and they live in water. They don't have seeds. They're propagules. Their spores and gametes are often cast out into the water, and they're naked. There's no sterile covering for most of them. And they don't have flowers, among other sort of classic land plant reproductive structures. And the reason why, we could spend the whole talk on this, the reason why algae are so hard to define with a very clear they have this and they don't have this is because algae have evolved multiple independent times across the tree of life. So the ability to photosynthesize has come about through a process we call serial endosymbiosis. This has evolved multiple independent times. And the very astute reason, or the thing that Kira alluded to with the land plant being an alga, is that the green algae, the red algae, the land plants, and another group called the glaucophytes are believed to have acquired their ability to photosynthesize from a shared common ancestor. And so if you put, everything that descended from that one ancestor into the same bucket, they're all part of the same big group, then if you're calling some of those algae, don't you have to call all of them algae? So it can be a little controversial, but as my advisor, Gary Saunders, would say, land plants are just a burp off the green algal evolutionary lineage. Okay. Where do we put moss? Kira, get out of there. No more questions about land plants. Okay. Oh, and someone asked, is H, so we go back up here for 1 sec. H is pretty dry. It's quite dry. Yeah, it's not wet and stringy in texture or slimy. You would mistake it for a lichen very easily. Okay, so I'm going to move on. So hopefully there's some sense that algae are this very broad, independently evolved group. That's why they're hard to categorize. The most familiar algae that we all tend to know and love are the big ones and the big marine ones. So seaweeds are a subset of the algae. Not all algae are seaweeds, but all seaweeds are algae. So we're familiar typically with the brown seaweeds. Here I got a selection of kelp. I'm going to come back to the name of the giant kelp in a moment, or in a few minutes, actually. We know the red seaweeds. There's a beautiful diversity of them here. The red seaweeds contain the highest number of species of all the big seaweed groups. There's the reds, the browns, and the greens. They each evolved independently of each other. So red seaweeds have their own origin, green seaweeds have their own origin, brown seaweeds have their own origin. And we have the most diversity of species in the red seaweeds. And then we're all familiar with probably with some of the green seaweeds as well. And that sea lettuce is right there in the middle. Okay. And when you go down to the beach, which is amazing, and this I took this in Banfield a number of years ago, you see them all mixed together. So reds, browns, and greens, you find them, neck and neck, sharing the same rock in the intertidal together. And so today I wanted to share a couple of stories, and I thought some of the things that are pressing on people's minds, I get asked a lot about this, even though this is not my particular area of study. I still, people want to know, we're all concerned about climate change. We hear about declines in all kinds of groups. And so people want to know what's going on with climate change and seaweeds. And so today I thought I would talk just about one sort of story around climate change and seaweeds, and I'm going to focus on the kelps. So in general, kelp forests, this community of all these different species, and the big 3D structural producers are the canopy-forming kelps, like your giant kelp and your bull kelp, creating these big, huge underwater forests. In general, kelp forests are declining rapidly around the world. And so here's a little story that came out of the CBC. It's about showing 2 photos of the same bay in Barkley Sound on the west coast of Vancouver Island, where some colleagues of Kira and I actually, so Sam Starko, I mentioned him a few times, and Chris Neufeld and others, documenting. You can see in the top photo, there's a bunch of sort of black things bobbing in the water. Those are the floats of a bed of Nereocystis, or the bull kelp. 2 years later, after that, the blob we had from 2014 to 2016, this extended marine heat wave, the bull kelp were gone, and they did not seem to come back. The most dramatic example of decline in kelp forests around the world really comes from the southern end of Australia, so Tasmania, and then the southern end of Australia, where they've seen 95% declines in their kelp forests, or even entire local extinctions of kelp. So they're just not there anymore, and they were a number of years ago. And so lots of other places have documented, these we're seeing shifts away from kelp forests in some cases to more turfy communities dominated by little filamentous seaweeds. Rates of decline seem to be increasing. And so there's, lots of sort of evidence to support that these forests are in decline. And one of the major, the major driver of this globally is warming waters, both gradually warming waters and then marine heat waves of higher frequency and greater duration and increased intensity happening kind of around the world as the main driver, and those being the result of human-created climate change. And so I'm just going to touch on this one study. So this is a summary, this figure showing the world and the coastlines, and a summary of daily coastal sea surface temperatures. Anywhere that's shown in red is showing sea surface temperatures that are above this 37-year average. So they're warmer than usual. A little example in a few places of cooler than usual, but predominantly warmer. And what this study really nicely tried to document was that different species and even populations of the same species, but in different areas, these kelps are responding differently to warming waters. So it can be challenging to kind of paint this all with broad strokes, because depending on the population, it may be holding steady, it may be declining greatly and be locally lost, or in a few cases, seeing actually some range expansions. That tends to be the northern end of ranges or northern species, and then things in the south or towards the equator, equatorial populations are the ones that are really declining and contracting. So just some take-home messages from this, that some kelp-dominated habitats are persisting despite increased temperatures in those areas. But a bigger pattern is that many others are declining or they're entirely gone. And where they're seeing the biggest contraction, as I said, or the biggest loss is wherever the range is that's near, equatorial regions, or the southern or northernmost end around the equator. Those are really being hit the hardest. But I wanted to bring it back to BC, and this talk today is so perfectly timed because a new study just came out from colleagues all over BC, including Julia Baum at UVic, Sam Starko that I mentioned. So Sam and Kira Janot, who's on our call here, were lab mates at the same time doing their PhDs here at UBC with Dr. Patrick Martone. I've had the privilege of working with Sam over the last few years. So there's this great collaboration going on, and there's all kinds of initiatives going on actually around BC in understanding what's going on with kelp populations and then turning that into conservation and restoration efforts. So this paper just came out documenting local and regional variation in kelp loss and stability across BC. So I'm going to walk us through some of the data from that. And just today, Sam, Brian, Timmer, Julia, and Chris Neufeld put together a for the public sort of summary of this paper and the conversation. So I have the link for that there. This is a really nice summary of the study. And what they are talking about and documenting is what is happening to Nereocystis and Macrocystis, our two big canopy-forming kelps in British Columbia. All right, let me know if I'm going too fast or if this is really confusing, but and I can slow down or I can reiterate. What I'd like to spend a few minutes on, because I think this is probably of top of mind for many folks in this group, is trying to understand what these researchers have put together in trying to understand what's going on in BC. Because one of the things that they point out in this paper, which is shocking to me, is a reminder that BC has a greater coastline in terms of kilometers than Oregon, Washington, and California combined because of the action of glaciers as they were receding. And yet our coastline in British Columbia is just tremendously understudied compared to what's happening on the west coast of the United States. So we don't really know what's happening to our kelp, or at least we didn't until this paper just got released. So they were documenting what's happening to these two kinds of kelp at these 11 different sites from the southern end of British Columbia to the northern end, and I've listed all the names here. And this colorful figure is showing us where in BC, based on lighthouse sea surface temperature measurement stations, where waters have been warmer than usual, and where waters have been holding steady in temperature, and where there's actually relatively cool waters compared to other parts of the coast. Okay, so I thought I would take a moment here to stop talking for a second and give you a chance to look at this. And then I'd love you to make a prediction in the chat if you feel so inclined. You know, make a prediction. We see red, so warmer waters, the warmest waters, 20 degrees, almost 21 degrees recorded sea surface temperature in the inner Salish Sea. Whereas up north in the outer coast, you know, summer sea surface temperatures holding steady at like 12 to 13 degrees Celsius. What they did is they took data based on aerial photographs of kelp beds, so either taken by planes or by satellites, whatever they could get together, and they looked at kelp coverage as far back in time as they could. So the earliest data points were 1997 in some places, 2007 in others, and then a more recent time point, 2019 to 2021 or 2018 in a couple places. And they compared those two time series, what was going on in the late 90s, early 2000s, and what was going on in the later, 20-aughts and 2020s. So I'm curious to know what you think, what might be happening to our kelp beds across the coastline of British Columbia. If you'd like, you can enter in the chat now and tell us what you're thinking, or put up your hand, anything goes. Or if you're too shy about it, that's also fine. You can just think about it. Joan says the Salish Sea kelps are suffering. Okay. Kelp disappear inside the Salish Sea, says Sheila. Yeah, we've just talked about warming waters and kelp declines. Okay, kelp's disappearing. Would you say the most inside the Salish Sea, Sheila? Okay, so kelp's disappearing the most. So there's a correlation between those sea surface temperatures being highest in the Salish Sea, or in fjords and inlets all the way up the coast, and that correlating with the greatest declines in kelp forest coverage. Okay. Any others? One more person want to contribute? You don't have to. Oh, Dorothy, what about sea urchins? It's like I planted you in this audience because we're going to talk about those in a second. Okay. This story is more complex than just sea surface temperatures. I'll give you that. Okay, let's go through it. All right, so we're going to walk through this. So I've started with one site, which is the Gabriola Valdez Island subregion, they're calling it. And so the way they're displaying these data, the summary of these data is they've got bars that run across. Basically, they're comparing these two time points for 2004 versus 2021 for Gabriola and Valdez Islands. And they looked at photographs and they basically quantified how much kelp there was in 2004 and how much kelp there was in 2021 based on what they could see in these aerial photographs. And red indicates some sense of the amount of loss. Light blue indicates holding steady, and dark blue would indicate gains in kelp coverage. I realize I covered up the little legend now with my labels. So I'm going to zoom in because they have these really beautiful figures that actually zoom in on the exact spots where they measured their kelp. So here are the two islands. Everywhere there's a circle is a place where they compared photographs for 2004 and 2021. If that circle is red, there was less kelp in 2021 compared to 2004. There are a couple of light blue circles, which means they were comparable in their coverage. And there were no gains. So in nowhere on those spots they measured did they see more kelp in 2021 than they saw in 2004. And this was the most extreme loss, I think. They estimate that 74% of the kelp coverage in this subregion was lost between those two time points. So if we come back out and we look at, okay, what they've called the southern part of British Columbia, which is Gabriola Valdez, Maine and Saturna, Cowichan Bay, the entrance to Juan de Fuca Strait, West Coast Trail, and Barkley Sound, there's variation. There's variation in loss, and in a couple of places there's actually gained. So they've seen an increase in kelp coverage in 2 places. So I'm going to zoom in again on some of these locations. So here's showing one figure showing all of those 6 southern locations grouped together, or excuse me, yeah, 6 southern locations grouped into 4 boxes. So they saw a little bit of gain in Cowichan Bay, which is very interesting and has yet to be, I think, fully explored and explained. But they also saw a little bit of gain in Maine and Saturna as well. And then lots of holding steady in around the West Coast Trail and the Juan de Fuca entrance, which makes sense because there's cooler waters there. And they measured a lot of places along the West Coast Trail. That must have been fun. I mean, I guess they didn't really go hiking. They just did it from photographs. So to summarize, for the southern part of British Columbia, the authors say that the summertime waters now exceed the maximum temp that these kelp can withstand. There's all kinds of literature now documenting the maximum temperatures in which these kelps thrive or can even survive. And sea surface temperatures inside the Salish Sea in the summertime, those summertime hottest days, it's now exceeding what these kelps can stand. The inner Salish Sea doesn't have Macrocystis anyway. There's some interesting ideas about that. But so Nereocystis in the inner Salish Sea doesn't like it as hot as it's getting. And so we've experienced large losses, and they're attributing this primarily to warming waters. If we come back out again and look at the central coast of British Columbia, so this is Laredo Sound and the south central coast, the X and number 9 and 10 on their list. This is where they've seen more significant gains and really holding steady. And something that the authors talk about here, which is really interesting, is that these kelp populations typically are, the majority of them are holding steady or even expanding in size. And these are also the parts of British Columbia in which sea otter populations have expanded to numbers enough to keep the sea urchin populations down. So they make that point that this is where the sea urchins are doing well. And then finally, I would have predicted that the northern part of British Columbia was maybe doing the best, but unfortunately that's not the case. Even though the waters in northern British Columbia are cooler and staying relatively cooler compared to the southern part of the province, they're still seeing quite significant declines in their kelp forest populations. And to come back to Dorothy's point, this is being attributed to expanding herbivore populations, in particular the sea urchins. And this is because the sea otters have not come back. And so there's nothing to keep the urchin populations in check. However, they also talk about some reasons for optimism. So even though our canopy-forming kelp forests have declined considerably, not all the populations have declined, some have even gained. And they really emphasize the importance of local scale factors influencing kelp fort responses to climate change or environmental change. So there's local variation in how warm the water is getting. There's local variation in how prevalent the herbivore pressure is. There's some places have the sea otters there, and so things are a little bit more in balance. And they also really advocate for protection and restoration of kelp forests is going to require what they call innovative solutions as opposed to passive protection. And so passive protection would be something like creating a marine protected area and keeping, excluding, say, harvesting, maybe not having coastal development near a marine protected area, but still that's really quite passive in its conservation work. They actually, they talk about, and this, I put the study here, there's a new study that also just came out that's really fascinating, that really well documents how marine protected areas are helpful for kelp conservation and restoration, but they're not enough. And it's literally called, the paper is that MPAs are useful, but not a silver bullet for kelp conservation. And they did all this work documenting what's happened in California. There's Norway, there's New Zealand. So there's like some global patterns they've been able to pull out. That's fascinating. And that paper, I believe, is open source, so free to anyone to access. And in Sam Starko and his co-authors, they are arguing that controlling urchin populations, and actually this is work that's going on here at UBC and other places, people are looking for kelp strains or variation within kelp that are able to tolerate higher temperatures better. And so maybe we can outplant with a more tolerant population of kelp to better withstand or even thrive in warmer waters is some of the thinking here. And I'll come back to that question in a sec, Sheila. I also just wanted to shout out this, there's a report here, I've got the link there, bringing together a number of collaborators from all different relevant parties, but in particular being led by Indigenous communities and Indigenous knowledge, is, I think, a reason for optimism. And up in Haida Gwaii, the Haida Nation has really been, I think, at the forefront of some of this work. And there's a report that documents these indigenous ethics and values leading to ecological restoration for people in place in Guayhanas. And part of this project is an attempt to restore their kelp forests. And so this is the part of the province where the otters have not come back. So there's tremendous urchin pressure. Sheila mentioned the sea star wasting disease is another sort of piece in this puzzle where other predators beyond otters, such as like sunflower stars and other sea stars, have been tremendously hit by the wasting disease, which is exacerbated by warming waters. And so you get even more explosions of urchins that then really impact the kelp. But so this effort going on in Haida Gwaii is really interesting because, and this is really being led by the Haida Nation, they're mimicking the presence of sea otters by removing the urchins in order to provide space for the kelp to then get a foothold and grow into a kelp forest. And so they have divers who go down with the explicit goal of basically smashing and removing the urchins in the area, clearing them out. And there is evidence that this kind of restoration practice is working to see gains in their kelp forest coverage. And so this kind of big collaboration between a number of relevant parties is thought to be another way in which we take a more active role in protecting and then restoring kelp forests in British Columbia. That was a lot. I wonder if anyone has a question there. That's going to be the end of my sort of like doom and gloom climate change. I want to get into a couple other things now. Sheila, I haven't looked up hydrocarbons. I'm gonna have to think about that for Saturday. I know I had that in my notes to look up for this talk, but I never got there. I'm sorry. All right, gang, I'm a taxonomist. I want to talk to you for just a moment about some taxonomy because it's important. But I know as an end user of taxonomy, it is also very annoying when a name changes. And so I want to give you a story and some rationale. So Murray asks, is the removal of urchins going to have to continue indefinitely? That's a great question. I don't know. I'm sure there will be cycles of removal, especially if otters have not returned. But I don't, yeah, I imagine there will be cycles, years where there would have to be a lot and then years where there would have to be less, but I imagine there have to be some sort of maintained management. Yeah, I imagine for at least the foreseeable future. Okay, So I get asked this, I mentioned this in the little blurb for the talk, that Macrocystis, our big giant kelp, until recently it was Macrocystis perifera. When I was younger, it was Macrocystis integrifolia. Now it is Macrocystis tenuifolia. So the name has just changed as of last August. And I want to tell you a little story around that. So our Macrocystis, well, around the world until 2009, there were 4 species of Macrocystis recognized globally. On the coast of British Columbia, we had populations of Macrocystis integrifolia and Macrocystis perifera, and they were distinguished by features of their holdfasts and blades. The holdfast is the really dramatic example. Perifera has a very cone-like holdfast. It's like a big cylinder. And then integrifolia has a more like rhizoidal, it's like a big creeping holdfast that's quite distinctive from the big cone one. A pal of mine, he lives in Port Moody, Kyle Demas, when he was a graduate student, one of his contributions to the phycological community was based on quite a bit of evidence at the time, it was proposed that actually those 4 species should be combined into one, and Macrocystis perifera was the older name, I believe, so it had taxonomic priority. And this was based on a lot of really cool data, including like interfertility between populations, plasticity, so the kelp holdfast might look different depending on where you grew it in the intertidal. So this figure here from their paper is showing that what's been documented in California is that integrifolia, that creeping holdfast, is most common in the intertidal, and the big cone one is most common in the deeper waters. Okay, so then in 2009, it all became Macrocystis perifera with recognized ecomorphs or local variation and population level variation. But now that we have much greater molecular tools at our disposal, such as whole genome sequencing, it turns out that the taxonomy of Macrocystis is more complicated than recognizing a single species. So it's been known for a little while now that you can actually really well genetically distinguish integrifolia from perifera. So they're genetically distinct from each other. Now that we have more genetic regions at our disposal to look at and compare. And so, and in addition to this, there's kind of a geographic break where the northern hemisphere integrifolia and perifera morphs and now genetic groups are actually genetically different from those same looking Macrocystis in the southern hemisphere. And so a colleague here at UBC and a very prominent phycologist, Dr. Sandra Lindstrom, has done the heavy lifting of actually reinstating a name to document the northern hemisphere, or at least north of Point Conception in California, Macrocystis populations. So right now, they're all Macrocystis tenuifolia. In the future, there might be a little bit more splitting of that up, but right now, it's all tenuifolia. And so that's where we stand right now, is tenuifolia is north of Point Conception in California, and then perifera, integrifolia, those names still exist, but they only apply to Southern Hemisphere Macrocystis. Okay. And I just want to point out, I spelled taxonomic wrong. These changes, I know they're annoying. I find them annoying, and I use them all the time. It reflects our evolving understanding of the boundaries and relationships between species. So it's not meant to just make life harder for us. It's there because they reflect our changing understanding of something complicated like the evolution and the history of relationships between these different species. Okay. So I want to shift gears a little bit now, and I want to talk a bit about the work that I do in getting people engaged with seaweeds and helping to educate others with seaweeds. And actually, some of the reasons I think this is important. So I'm an educator now full time. I do research, but it's research on teaching and learning. And I have been teaching about seaweeds for a long time, or algae more broadly for a long time. At the same time, I'm a human in the world, and I understand and I see evidence of how the science of biology can be misused in social ways to discriminate or oppress people, and so I really embrace bringing diversity into my classroom and inclusivity in my classroom and equity in my classroom in all forms. And this paper that I'm showing up here is this really beautiful sort of summary of a whole bunch of work on how post-secondary biology classrooms can embrace gender and sexual diversity and actually more accurately reflect the diversity in the natural world in a way that then isn't overly simplified and then misused or misunderstood by people and applied to human social constructs. So there are these 6 principles that these researchers came up with. I'm just going to touch on the top one there, that present diversity first. And if you're curious about this kind of thing, the same folks who put together this paper are the ones who have this beautiful project called Project Biodiversify, where they're soliciting teaching materials that really show the people behind the science at every career stage from all around the world, from different cultures, because science and other ways of knowing about the world exist outside of that sort of more classically or the gate-kept Western science perspective. And so this is to celebrate the people who are actually doing the generating of new knowledge that moves the world forward. I've submitted one person to this repository, a little teaching guide to the work of Dr. Isabella Iona Abbott, who happens to be an incredible phycologist. She passed away 12 years ago now, I believe. And Dr. Abbott did a lot of her work in California, but she grew up in Hawaii. Her mother was a native Hawaiian, and her father came from a Chinese background. And so she brought ethnobotany into her science and also described over half of the marine flora in Hawaii. So just, as a way to shout out this person and their tremendous work, I put this out there as a teaching resource. And so one of the principles, and this is one that I've really taken to heart, is in your biology classroom, you can teach topics sort of from a human first or even human only perspective, because often we're pressed for time, or we don't want to have to explain things, or we don't really understand other groups of life well enough to feel comfortable sharing that in our classroom. So we focus on humans or animals perhaps. But the advocacy here is in presenting more diversity. There's more than one way for cell division to occur. There's definitely more than one way for a life cycle to occur. All these kinds of things that then often get misused, either accidentally or on purpose, in a social construct. And so I tell everybody that algae, seaweeds included, are amazing for teaching about diversity because they do reproduction in all kinds of wonderful and wacky ways. Their sexual life cycles are just, there's all kinds of, every kind of sexual life cycle is present in the algae. There's dimorphic sex, so males can be distinguished from females, but there's also intersex. There's monoishi and dioishi, which is when an individual, monoishi is when one individual contains both male and female gamete producing structures. Like, you name it, it's in the algal world, and is great for showcasing how the way that animals do things is definitely not the only way, and it's also not the most typical way. Okay. I also love that this paper really highlights that there's a ton of opportunities for meaningful education and outreach. So they note that there's a great need and demand for more ready-to-use educational materials that illustrate examples of, in this case, sexual diversity across the biological world, and they want to see those things developed. So this is kind of like my mantra for what I do with a lot of my students. Add to this that, as Joan said at the beginning, I'm a mother. And I gotta tell you, I'm really sick of just all the animal examples in children's books and activities and TV shows and on and on and on. Seaweeds, and more broadly, photosynthesizers, are just missing. And these are my 2 children. Miles is on the left and Cora's on the right. These two unfortunate souls are just inundated with seaweeds all the time because of their mother. But if I can change their minds about how the world works, then I guess I've done my job. Which brings me to my teaching here at UBC. So I am a big advocate as an educator of doing things in my classroom that can leave my classroom and have value outside my classroom. So I want to tell you about two projects that my third-year biology undergraduate course about algae work on. And so the first is a project that my class does every year. The course code here is Biology 320. It's called the Survey of the Algae. And every year now we do a project where we document the diversity of seaweeds somewhere in Vancouver. It's been 2 years, and so far we've picked 2 spots at Stanley Park. I want to go to Kate's Park or somewhere on the North Shore, but I have as yet to build a strong enough relationship with the Tsleil-Waututh in order to have permission. So for now, we're staying on Stanley Park. This is last year. Some of the students, I've got 60 students in the class each year. And this is a good chunk of the students. We were just out a couple of weeks ago, and Sheila actually joined us for one of our trips. So one of the ways in which I get my students to make contributions outside the classroom is to document diversity through the community science platform called iNaturalist. And this is our project page for this year. So the link is there, and you can go and see. At the moment, we're just wrapping up, and right now we have 183 observations of 39 species from 2 trips over 1 weekend in March. So I'm a big user of iNaturalist. I think it's a wonderful platform. So much amazing documentation of our natural world is on there. I gotta say though, that seaweeds are not an algae writ large. The observations are often not great. And I think with a little bit of tweaking, people's observations can be made much more useful. This, I just grabbed one I saw recently that came from, I think this is at White Cliff Park. It's a nighttime photo. It's supposed to be a red seaweed. It probably is, but I couldn't get any farther than that. And I know my seaweeds, and so does Kira. I wonder if Kira has any idea what that is. It's very common when you look up iNaturalist observations of the different seaweeds for them to be of poor quality to the point that you can't really confidently say what you're looking at, which I think is a problem. We need to better address making these observations so our data set is robust and actually useful for documenting diversity. And so I'm saying all this today to this group because I'm hoping there's a few iNat users in this group. And I would love to sort of not only encourage you to do the city challenge coming up, but take a few extra moments to really sort of capture the seaweeds well, because they're quite underrepresented relative to like birds, which are like everywhere. I'm just kidding, birds are great. Okay, so I want to feature a couple of the local seaweeds we have in Vancouver, and when we go on the weekend, this is one of the beautiful beasts we're going to see. This is called Hollenbergia subulata. It's a very finely branched, we call it filamentous because the branches are as thin as a human hair. They're so small. And so that's what it looks like at the beach on the right. It's a big clump of hair-like filaments. You get it under a microscope and you really see the beauty of these finely branched seaweeds. This is a record made by one of my students this year. And so there are very few records of this species in total for the whole world. There's 23 iNaturalist records. 20 of them have been made by my students in the last 2 years. One of the features that you need to tell that this is Hollenbergia is you need to see clear gland cells and where they're placed. And that little yellow arrow is pointing to a gland cell. It's at the end of a branch. That's how you tell Hollenbergia apart from a relative called Scagilia. If you don't have this information, you cannot call it Hollenbergia, or I would not be able to confirm that it's Hollenbergia. Something else I'm really proud of my students documenting, these are sperm-producing structures. All those are like little frilly white comb-like things. Those are the sperm-producing structures on another kind of filamentous red seaweed called Polysiphonia. My students found this at Stanley Park. I've never seen sperm-producing structures in the wild. They're so hard to find for Polysiphonia. And so now this is on iNaturalist, so we can explain what these are and how cool they are. Joan asks, do you need a microscope for IDing many seaweeds? Sometimes yes, but not always. Some things yes, but not always. These very finely branched reds, a good hand lens is often enough, actually. So in addition to iNaturalist, our students are making collections. The pressed seaweeds that they make in the lab are actually going to Vancouver's Natural History Museum, the Beatty Biodiversity Museum at UBC. Here's a selection of seaweeds from last year that are now in the Beatty Museum and part of our record of the flora. And these are a few that the students made this week, documenting some of these seaweeds. Joan, you wouldn't need a microscope. Even that one on the top right, if you look closely enough, you'll see the way it branches, and you'd be able to get it down to two things, Scagilia or Hollenbergia, so you could get really close without a microscope. All the other ones, though, you can ID without a microscope. Interestingly, documenting of local diversity has really declined in the last few decades. We had sort of a heyday of a few phycologists who were here at UBC or in the Lower Mainland, and they were documenting seaweeds like crazy around the Lower Mainland, thousands and thousands of specimens in the Natural History Museum or over in the Royal BC Museum, and that has just dropped right off to the point where a lot of things just are not sort of counted in the flora regularly, even though we're in Vancouver and there's a ton of people doing this kind of work. I wanted to shout out a couple of examples of beauties that I think we're going to see. So this is Opuntiella californica, or red Opuntia. It's a really beautiful red seaweed. It's got these big loby branches. It's this beautiful deep red color. There are relatively few. There's like 90 in the whole world and 36 records for BC and Puget Sound. Some of those have come from our class. This beast likes low water, so you have to have a good low tide. Our low tide on the weekend should be possible to see this. But it's a really beautiful seaweed. And the Beatty Museum hasn't had a specimen deposited of this species of seaweeds in 41 years from the Lower Mainland, and not from Stanley Park in over 70 years. And so our Natural History Museum, like maybe people have noticed it, maybe it's even been in a paper or two along the way, but if we don't document these things in our natural history museums, there's really no way to go back in time later and see what was in our flora. So I and others are really advocating for a regular documentation of our flora going into a museum where these specimens, if all goes well, will be around long after we're all dead. So these are contributions that our class is doing, and I think groups like Nature Vancouver can make contributions like this as well. Anyone can make a submission of a pressed seaweed to a place like the Beatty Museum. Okay, good question, Tatiana. I'll get to that in a minute. Another one I want to shout out, I call this Vancouver seaweed because Sparlinia pertussa is a beautiful red. It's full of holes, and that's developmental. It's not herbivory. And again, hasn't been documented in a long time. You can't find Sparlinia in very many places. I don't know why it loves Vancouver, but I had never seen this before except one place in Bamfield around the entire island. So it really likes it here. It's beautiful, and I think we should celebrate it. In case you were thinking those little red guys, I mean, actually Sparlinia can be like this big. Our kelps are also not being recorded very well in our Natural History Museum. So we were making collections that were the first in 30 and 40 years of our bull kelp and Alaria, which is our winged kelp. Okay. Is there anybody here who uses iNaturalist? Can I get a sense in the chat? Because I have a few slides, but I can also kind of skip this if people are not. I've kind of made a real effort this year to talk to some experts about iNaturalist use and how to really make strong records, also, you know, based on my personal experience. Okay, so maybe these slides are useful. I realize I'm already like an hour in. Okay, maybe I'll share my slides on this because I was really rigorous this year with my students, and so I am. Oh, Alan, do you have a question, or are you just putting up your hand to say yes to the? Okay, good. All right. My timing is fine. Great. The question about field guide I'll get to in a second at the end. All right. So this is what I've created for my students this year. And just as a general I'm going to give you some general advice for how I think anyone's iNat records can be improved. And it has a lot to do with which photo you choose to put first. So it's the display photo, and what information you choose to put in the notes fields. This is especially important for groups where there's not a lot of options for the attributions, like what life stage it's in, and etc. So the first photo, if you're going to put like 7 photos in an iNat observation, because you took one, you know, in your hand and you took one with a ruler, blah, 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 all these options you can have for variation, that first photo really should be diagnostic, because people don't really, it's hard to know that there's multiple photos to go and look at. So in my case, if I have a frilly red seaweed and I need a hand lens photo or a microscopy photo, that's got to go first. And then for us in the seaweed world, you need to know what stuff is growing on, or if it's drift. You really need, it's good to know if it seems common or if it's the only one you saw. It's nice to know if it's reproductive, but that last line of which features led you to pick the name you picked, that's key in my mind. And then the question is permission to be accessed by GBIF. I'm so glad you asked. I'm going to put on my science hat for 1 sec here. If you haven't heard of GBIF, it is this phenomenal international effort to provide basically a clearinghouse for biodiversity data. So it stands for Global Biodiversity Information Facility, or GBIF, it gets called. And so you can go there and you can look up Nereocystis, luetkeana, or any species on the planet, and GBIF pulls from all these other sources and creates this little like curated list of observations for you. It's pulling iNaturalist records, barcodes, so DNA barcode genetic records for many groups. It's pulling herbarium or other museum specimens. And there's a couple other data sources depending on the taxon or the group of life you're looking at. So you can see all this data in one place. It has a fantastic mapping tool for looking at things in a specific region. I highly recommend people who are curious about biodiversity go and play around with GBIF. It's got great tutorial videos. It's an amazing resource. And so scientists use GBIF a lot and ever increasingly to look at biodiversity data around the world. And to answer questions about biodiversity and things like phenology, like changing flowering times or changing seasonality. They'll pull records from GBIF to answer some of these questions. You, as an iNaturalist user, can take a few small steps to make sure that your records on iNaturalist get pulled into GBIF, because GBIF has quality standards. So if things fall below a standard, GBIF won't pull them. And so your data, it's great on iNaturalist, but it could be even better and more visible if it was getting pulled into this large clearinghouse database. And so I've learned a couple of things about this. And these are the slides I used. I like sat my students down and we had these slides up and then they all did these things. So one of the things you need to do, if you're curious about this, you as an iNaturalist user, you set the copyright permissions for your observations and for the photographs of your observations. So essentially, if you go into your account settings, there's a spot where you can choose the licenses, and I have a link to a great article explaining licenses. They have really nicely put little GBIF icons. So you make sure that your observation license is set to one that has the green GBIF box, and you're good to go. You can edit individual records, so you can change a license for a certain record in a different way. I can talk about that if anyone's curious, but if you want to just say, all my records should be GBIF compliant, you go into your settings and you change it there. The other thing that you need to do if you want your records in GBIF is the geolocation accuracy. So basically the size of the circle around where you say you collected your specimen, it has to be under 25 meters in order for GBIF to pull the record. So if you didn't have location services on, or if your phone was sort of wonky and it was like, oh, you picked this up somewhere in 172 meter radius, GBIF won't pull that record, even if the license is set to do it. So if you're like me and just learned this recently, you can actually go in and edit all of your observations at once. So these two slides are just what I had my students do, is you can just go in and say, okay, for all of my records, I actually want the accuracy to be below 25. So that means the circle is nice and tight, meaning that, you know, the area in which you say your specimen came from is just smaller than like all of Stanley Park, for example. Okay. So these slides are maybe kind of chunky and a lot, but that's, you know, I basically built these little tutorials so my students could edit their posts. Okay. The last thing I want to say about iNaturalist, for those who are here, so, you know, this is a post that my students made, one of them before we talked about iNaturalist and GBIF and all of that jazz. So here's a beautiful observation, but to make it more useful for science, they needed to do a couple of things. First of all, that photo is nice, but it's not diagnostic. They need to show, in this case, the gland cell on the little filament. So they have that photo, but they need to put it first. Otherwise, a user has to kind of go in and dig around to even know that's there. So put the photo first. They had copyrighted their photo and their observation, so that's not useful. So they had to change their permissions. And then all that information, I put it in the notes fields. They needed to flesh that out so that I knew what features they were using to say that it was a Hollenbergia, where it came from, etc. There's a fantastic article. It's on the iNat journal forum, I think, where one of the technicians actually went through and explained all the licenses for a general audience and why it's important to use GBIF, etc. So that's there for anyone who's curious about that. And then the last, if I still have a few minutes, the last thing I wanted to sort of highlight here, and this is, we're actually having our showcase tomorrow, so I'm so excited about this, but I'm also really nervous. The other big project that we do as an algae course that leaves the classroom is they need to create something that's going to educate their friends and family about algae. So sometimes they pick seaweed, sometimes they don't, but it's an algae course, so they have to create something that's going to leave the classroom. Last year, this was our showcase. There's 60 students. We had a whole bunch of people from the museum and other places come in and support the students. So I've never seen students put so much effort into bringing their science into a new space. We have seaweed themed Guess Who games. You know, that game from like the 90s is when I played it. So basically you're guessing your alga. One student was she's really good at designing jewelry. So she literally built a chessboard with seaweeds embedded inside, and she had to figure out how to preserve seaweeds in resin. It was this whole process. It was amazing. This is sitting in my office now. I take it with me whenever I talk about how cool seaweeds are in art. We've had seaweed themed Uno. A student who's Lydia, she actually designed crochet patterns for 2 different seaweeds. It's free to use and share, and I have resources to share with you all at the end. That's peppered with fun facts about these different seaweeds. So you can actually crochet your own bull kelp. We've also had a bunch of student-created children's books that are free to use and share. I have a small grant to print copies, so I have a bunch of them in my office. We've taken these to several schools and read them to kids and did seaweed outreach. I am so sick of animal-themed coloring pages, so a bunch of students have created seaweed-themed coloring sheets that are actually local, accurate, realistic seaweeds. I love these so much. I think we have 12, and there's another 20 or so that students have made that they're showing off tomorrow. So again, these are free to use for anybody. They're Creative Commons license, so people can use them and share them and change them with attribution. I have a Seaweeds of Biodiversity poster celebrating our diversity. This is a collaboration with the Beatty Museum and the Botany Department. This is a PDF that you can have if you want to print your own, but we also just put it together as a template so you can replace the photos and make your own using that kind of cool, wavy, leafy design. And then a couple last things. It's the greatest joy of my job is that I get to do this outreach now with students. So as I said to some of you who were here on the call at the start, some of my students from this year's course are going to come with us on these walks to share their knowledge. Last year, there were 11 of us in total who participated in a Indigenous youth camp that's hosted every year here at UBC. I've been to my daughter's school. I've been to other schools. So we're just like really a little pod of enthusiasts who are bringing these seaweeds into the classroom as much as possible. And one final project I want to shout out is a recent collaboration with the BD has been, again, with students, is to build this new Explore Algae website where we've done a ton of work to put together annotated photographs of a bunch of different local species. This is the bull kelp as an example. We've labeled all the parts. In the captions for the photos, we explain what the parts are. We've tried to come up with phonetic spellings for words that are probably unfamiliar to most folks, like sporophyte or even Nereocystis. Like, how do you say these things? I hired 2 students. We've done 11 life cycles that are hard to access, but now we have free to use life cycles that are accurate and vetted by experts. So these are available on the website. And then many people probably don't know that our big kelps alternate between the big form you see and actually a microscopic filamentous stage. It's critical to the life cycle. Most people don't know they exist, and you will have never seen them in the wild because they're too small. So we're trying to put photographs and information out there as well. And again, so I get students, this is Nola and Maisa. Nola drew a lot of the life cycles. Maisa took a lot of the photographs, and we shared our website with about 200 educators from around BC at the museum's open house in October. So there's a whole bunch of stuff there. I know someone asked about where to get this stuff. I have a QR code and a URL at the end. I just have a Google Drive folder where I put all this stuff and people can just access it. That's all the information I'm spewing you. I know it was a lot. If for some reason you're keen to know more, my favorite time of the year is the weekend that I get to go to Ucluelet and partner with the Raincoast Education Society to just play with seaweeds for 3 days. The outer coast flora is out of this world. It's literally steps away from the Ucluelet Community Center, which is where we do all our work. It's a highly accessible beach. There's a daycare that just goes out and plays there too. Like, it's idyllic. We press seaweeds for people to take home. We make cyanotypes, which is an amazing form of printing, the first kind of, one of the early kinds of photography. So we do some blended, art and science and natural history and all that jazz. So if you're curious about that, there's more information there because it's the best weekend of my whole year. And then someone asked about field guides. So gang, I got a little nutty. And I built us a Stanley Park field guide. This is 2 of the 4 pages. I'm laminating these tomorrow. There's some fabulous monthly seaweed survey data for Empress, the site at Stanley Park, that's been done by 2 graduate students who are super amazing here at UBC. They've been documenting when are the kelps reproductive, what seaweeds are they finding every single month of the year for the last 2 years. So I put those data and with my photos, so we have I think, as regionally specific as you can possibly get, ID guide for this beach where we're doing our walks. So I'll bring those with us tomorrow. No, not tomorrow, Saturday. And with that, I'll wrap up. I hope there's some time for questions. There's the URL and a QR code for all the teaching resources I mentioned. I have a ton of collaborators and all my students who do this. I have a few grants. And I want to say thank you again for inviting me and giving me this chance to just talk at you for so long. And yeah, I'm happy to take any questions or just hear stories and chat with you. Thank you. Oh, and I will say, someone asked at the beginning where I got my t-shirt. I'm going to look at the questions in a second. I have no affiliation with this little business, but there's a local business called Owl and Bear Studio, and they make the most amazing natural history themed, biodiversity themed things, including a seaweeds of local seaweeds of BC t-shirt. Okay.
All right. That was absolutely awesome, Bridgette. Thank you so much. You've covered so many subjects in that time. So that's what I was saying. Yeah, just keep going, keep going. It's great. This is great. And I see one of your collaborators there is Linda Jennings. So she's doing a talk on plant pressings for the botany section in a couple of weeks, well, actually almost a month, I guess, until she's doing that. Correct me if I'm wrong. May 2nd, is that correct? Is her talk?
Yeah.
Yeah,
Yeah.
the second, it starts at 7 o'clock, not the usual 7:30, and there will be a display of various plant pressing equipment and samples.
Hey, wonderful. Thank you. So Bridgette, would the same process that you use for pressing land plants also work on seaweeds?
With modification, the way you press a land plant is similar to what you do for a seaweed. You need to get them dry quickly. Well, you need to press them flat. And I see, sorry, I just want to say, Donna Gibbs, I neglected to mention the Seaweed Sorter app, which is my bad. My colleague and collaborator, Patrick Martone, created the Seaweed Sorter app. It's, I think, only about $5. We actually buy it for all the students in our course. I use it all the time. It is amazing. So if you're interested in an app that helps you identify over 110 local species of seaweeds, I cannot recommend the Seaweed Sorter enough. It's on iPhone and Android. Yeah, and we'll have it with us at the field. It's amazing. So Seaweed Sorter app is a great resource. The pressing question, so seaweeds are wet. So in order to press, I should have put I have a little cheat sheet for my students about how to press. The seaweeds have to get flat and they have to get dry. And so we use blotter paper and cardboard. Corrugated cardboard has those little wavy bits in the middle that allow air to pass through. And so you basically have to make a sandwich in which you've got your paper with your seaweed on it. And then around that, on below and above, are some sort of thick absorbent paper. We use blotter paper in the herbarium, but you can use newspaper in a pinch. That's what I always used in the field. Those like really stretchy blue absorbent towels for like car shops and stuff can work in a pinch. And then on top and below the blotter layer, you need something that allows airflow. So that's why corrugated cardboard is ideal. And then you put a fan next to your stack of presses, heavy books on top to really weigh them down, a fan to push air through, and then they need usually hours to dry. So different than land plants in that you're not worried about rot setting in because they're not very wet usually. One thing I'll highlight too is, and this we'll see this at Stanley Park. Those top two pictures on top left, they're kind of angling towards each other. Those are two examples of the acid weed. There's three different species of acid weed at Stanley Park. They're so called because they build and store sulfuric acid inside their cells. And so as soon as they're out of the water, they start to rot very quickly. They will rot everything else in your bucket, and they will not press very well unless you're really fast at getting them dry. So these were amazing presses done by students because they had to be really fast. Okay, what am I missing? There was
Murray, well, first of all, he said, thanks so much. Of course, it was an amazing talk. But he said while you were talking, he was looking at the Pacific seaweeds and noticed that some of the species you referred to weren't in your guide. I assume he's talking about the big book. Yeah,
so my book, Pacific Seaweeds, it's a guide to the seaweeds of the West Coast or Pacific West Coast or something. I forget the title. I wrote most of that book. Well, so Louis had the original version that he wrote many years ago in Bamfield. And then I was brought on as a collaborator to expand it. 8 years ago is when we published our new version. We added a bunch of new species. It's supposed to span from like Point Conception up to Alaska. And so there's about 640 different kinds of seaweeds that we know of in that range. And we only have like 120 or something in the book. So we had to make edits. Thank you. That's my book. Yeah, A Guide to the Seaweed. I can't read that, but that's my book. Oh, you have the kind that has the little metal we want on there. So we had to be selective in which ones we picked. So we were trying to pick a diversity of things. And so some of the things I mentioned today, like the two bottom seaweeds in this picture, that's Sparlingia pertussa on the left, and on the right is Constantinia subulifera. I had never seen those before, or very rarely when I was writing the book. And so I thought that they were not very common. And I think they are not very common, but if you're in Vancouver, they tolerate the low salinity of the inner Salish Sea. And so we see them a lot here, but we don't see them a lot in other places. So, regional floras or local floras are variable, which is why we don't have those in our book. Yeah.
Okay. So just thank you for that. A comment from Ben Gould saying that iNaturalist also accepts field sketches. So I guess if you have if you're a good field sketcher. I was wondering the coloring sheets and then you put up that the nice little
Oh yeah, I can put it back.
yeah, are they also on that site?
Yes. So all of the kids, so like there's PDFs of 4 of the children's books. These are the ones that are the most vetted and used, and they've been well received in the classrooms we've taken them in. So you can have PDFs of these. These are free to use and share. So students have agreed to let their work be used for educational reasons. The coloring sheets are all there. That big leafy poster of the seaweed biodiversity, that's there. The crochet pattern is there. And in about a month, after I go through permissions with the latest round happening tomorrow, there will be a whole bunch of other stuff, including books, more cards, and some other stuff. Yeah.
Wow. Yeah, that's amazing for you and also for them to be sharing all of that with everybody. You know, that's very generous. A question from Bev: do leather sea stars eat urchins? So she's noticing around Saturna Island, they seem to be expanding in number. There's now way more leather stars than purple sea stars. Where we see the leather sea stars, there were many young bull kelp.
That's a question for Sheila. I don't know anything about sea. The more I learn about seaweeds, the less I remember about animals. It's just, there's only so much storage space.
Actually, Bev, thanks. I did get that email and I haven't had a chance to look it up somehow. No, I'm not even going to guess. I can look it up and get back to you. Sorry.
Okay.
And I'm lucky if I can ID a leather star, so.
I wanted to mention that another good app if you want to for pictures of seaweeds, doesn't have any nice key or anything. The free BC Wildflowers app, among its 37,000 plants, includes I think 147 BC seaweeds.
Wait, which app?
BC Wildflowers.
Oh, who made that?
It's all the links and everything for it, and the best tips on best ways to use it are on the botany page of the Nature Vancouver website. But although it's called BC Wildflowers, it's basically all kinds of plants. It has lichens, mosses, and 147 BC seaweeds.
That's amazing. Well, I learned something. I mean, I've learned a lot of things already, but thank you so much for sharing that. I'm gonna go check that out.
They're only keyed out to green, brown, and red, so not by the number of petals or lobes or shape of their holdfasts or anything.
That's okay.
But it's great that it's available anyway. Oh, the other question I had is, when they're doing a herbarium specimen of something like a giant kelp, like something that's 20 feet long or whatever, is it like done in 20 pages, or is it kind of like folded and dried, or how are big, because seaweeds are kind of different, like most, I mean, I guess land plants, I mean, I guess it would be like trying to press the giant sunflower from root to top, but just wondered how it's done, because they're thin, whether you could just kind of fold them and do them on one page, or do you have to do 15 cards per plant?
It's a great question. And different people do different things. So I was just going, because we had from Brockton, we had an ulva that was, I've never seen an ulva this big. It was massive. And so the students spread it across 5 different herbarium sheets and then made cuts at the edge of the paper. And so then every specimen gets its unique field identifier, a little code that they make. And then when the museum will eventually add its own accession number. So the 5 sheets are just that code A through whatever the 5th letter is, E. That's one way to do it. And last year we had a Desmorestia herbacea, the big acid producing, the big strap-like one. That took 7 sheets of paper to get the whole thing. One when you're talking about a 20-foot kelp, most often people, if they're they won't preserve something that large in the first place. So there's a real bias towards smaller things in the herbarium. And I don't think that's unique to our herbarium. If they do try to preserve something really big, they will chop it usually. But they really rot because they're also very thick by that point. You know, you've got 100-foot bull kelp, and you've got the cortex and medulla and meristoderm, like the tissue layers are something like a centimeter thick. I mean, Kira could probably say more about that, but is quite thick. So it's hard to press. So yeah, we've documented that before, that like there's a real bias towards preserving things when they're smaller.
Thank you.
Great question though.
Can I jump in just for a second here, Bridget? I went to my faithful Rita O'Clair huge binder on marine invertebrates. And leather stars, for those interested, are the most carnivorous, and they eat whole encrusting sponges, bryozoans, sea anemones, sea urchins, sea pens, cucumbers, tunicates. It goes on. So, Bev, I think you have your answer there. Thanks.
So the leather stars at my place don't seem to be making a dent in the and all the green urchins all over the cliff in front of me here.
Oh.
But you know, maybe they're working on them. Another question from Alan. He's saying thank you for bringing the field guides, and he's saying for amateurs like himself, what would you suggest reading before then to learn the fundamentals of identification?
If you had access to the Seaweed Sorter app, in my experience as an educator with seaweeds, when you get to the beach, it's often very overwhelming to try to see one thing in a sea of a bunch of things. The seaweeds are all flopped over on each other, so it can be hard to sort of like see one. And it takes a while to build a search image for seaweeds. So I've noticed that the more times you sort of like look at them, you kind of get this search image so that when you sort of see something similar at the beach, you can kind of see it and distinguish it from the things around it that they're all flopped all over. So if you had a way to sort of flip through a picture book or a picture guide and kind of just build a search image, if you're a real, like if this is a novice experience for you, that's probably where I would start. I don't know how much reading you need to do necessarily. We're going to talk about things at the beach. I wouldn't worry about having like a ton of homework, but if you have access to a field guide, you know, all of those things have nice little intros to ecology and body parts. And so I guess a question for you then is, do you have access to anything like a field guide to look at?
The BC Wildflower app would be one that you could download and for free and have, and at least you could look through some of the differences.
Yeah. Or also that the website I mentioned, the museum website. Oh, there's another one. Oh my gosh, you guys. Does anyone know Josie Iceland? Have you heard of Josie Iceland?
Yes.
So Josie, this is her most recent book, The Curious World of Seaweed, and it's a beautiful mix of the art and the natural history and sort of the different stories of people, like who was Captain Lutka, for whom Nereocystis luetkeana is named for, you know, just like cool stories around where these names come from. And she has, what's her site?
Oh.
There's a new website. It just launched. She's probably going to win this award that I'm trying to win for my students too, actually, because her website is way fancier than ours. So there's this new resource called The Mysterious World of Bull Kelp. I think the URL is bullkelp.info. So it has lots of cool stories and some like cultural connections and some ecology. So that's a place for free, of course, you can just kind of explore a little bit about bull kelp and learn about them. But there's actually other species in there too. There's a bit about Macrocystis and some others too. So that's another place that it would be fun that kind of futz around to get ready for the weekend. Oh, but then our website is also, I'm really bad at this. Am I still sharing the screen?
Yes.
So here you can also read a bunch of stuff. I mean, some of the little tidbits in there come from Josie's book because we reference it. If you wanted to see what herbarium specimens look like, we're bringing in the museum. But if you want to learn what it looks like, we've got some, photographs I showed before. We've even got some that sort of feature epiphytes or things that live on bull kelp. And then if you want to, this isn't really for the beach, but if you sort of want to get a sense of how it makes all the way through its life cycle, we've got some information there. But we have this for Alaria, Nereocystis, we've got Maziella and Pyropia, and then 2 greens as well. So there's another place you can kind of dig around and learn some stuff.
Quick question on the green algae. Can you accurately even discern whether a green algae is 1 cell or 2 cells just by the color, one being much more of a spring kind of green as opposed to a darker green? Is that real, in terms of layers?
Can you tell how thick a seaweed is by looking at it, by the darkness?
By the dark or lightness of it, or because if it's 2 layers, I find it really difficult to distinguish. Like that's quite a light color, but I've seen ones that are very dark green as well, and I'm just wondering, maybe.
So there's Alva is 2 cell layers thick.
But
I would say no. So we don't have it in Vancouver, but there is another thing that looks just like Alva, but it's not. It's called monostroma because it's only 1 cell layer thick, and you would not be able to tell that apart just by looking at them.
So are you saying then that most of the broader leafed alga that we have locally are in fact 2 cells thick?
Yes.
Okay.
Kira's here. Kira, you're still here because Kira has actually done a lot of genetic work recently to talk. I mean, I shouldn't say anything because it's not published yet, but just I don't think we don't have monostroma in Vancouver. It was pretty common on the East Coast when I lived there. So you'd have to section things to figure out if it was alva or not. And then there's another one called alvaria, which dyes the paper black. So that was one way you could tell if you had alvaria or an alva, is you dry it. But around here, it's there. We have a ton of alvas, but they're all alvas, I believe.
Okay, if I find some where I can see similar size, but that very distinct difference in darkness versus that limey light green, I'll try and grab one. I feel like I've seen it, and it's always been something that I've wondered about.
Let's see, and you're thinking specifically of alvas. They start to go a little funky in their color when they're about to release spores or gametes. We actually had a bunch of alvas come back the day we were out, Sheila. When we got back to the lab, the bag, all the water was green because the alvas had started to release all their propagules. And so this was a bag full of gametes and then whitish alva, but they kind of go yellowy in my experience right before they're about to sort of release them. And I wouldn't say, so alva, like 1 to 2 cell layers is it's not much of a difference. So I feel like in my experience, I wouldn't tell 1 from 2 cell layers very well. And when we go to the beach, there's actually, so on the right, there's Pyropia. Pyropia is 1 cell layer thick. And at the beach, we're going to see Wildmania or Wildmania. And that one looks just like this, but is 2 cell layers thick, and you cannot tell. Like the difference between that many layers is just so subtle. But a Pyropia, you can see your fingers through it, versus Maziella on our iridescent seaweed. This beast is like many cell layers thick. You can't see your fingers through it. It's really rubbery. So like once you get between the thin seaweeds and the thicker seaweeds, you can tell by feel and look with those.
Okay.
Not with all those.
Bridget, on your green the ova one,
Yeah.
one of the photos showed a leaf that looked very opaque against the hand.
Yep. This one?
The lower
Huh.
no, the lower one in
The middle.
the middle of this slide.
Oh, this one.
So
That one's
the previous slide.
This one?
Yeah, in that
Hey,
photo,
oh, you were saying opaque. Yeah, that's the
Look-
opaque.
it looks quite opaque there.
I think it's the light. I was in shadow. I don't think it would definitely not be a difference in thickness. I think it's the lighting that's making it look more opaque than it actually is. And it's kind of like ruffled above my hand, so it's not really like flat against my skin.
Because actually, when you pull back, that's a good point, Teresa. The one with your hand, the other one that you showed that was very thin, both in the upper and on the left side. Just
Yeah.
see, that to me looks like the really limey green versus the dark green. But I guess on the beach, it could be that I'm standing over top of it or something like that.
Or it's like bunched up. I think there's maybe even like sort of a folded over part in that the photo of the one that's on the limpet. All the ones we've sectioned around here are 2 cell layers thick.
Okie doke. Thanks.
Even like, and then you mentioned the multiple sheets, like here's a big beast of an ulva that took 2 sheets in this case. And even this size, there's still 2 cell layers thick.
Okay.
Okay, well, we do have to let you go